Hello to all, myself Uma Ragiv, and in this video, I'm going to discuss about cell injury. I will going to cover the introduction, causes, pathology behind this and the types of cell injury. So we'll start with the introduction first. What is cell injury? Cell injury is defined as a variety of stresses a cell encounters as a result of changes in its internal and external environments. It leads to change in cell structure, metabolism, physiochemical properties and function, which leads to impairment of its vital capacity. So as you can see in the diagram, the normal cell, the ultimate goal of every cell in our body, is to maintain homeostasis. However, in certain situations, due to changes in the microenvironment, cells undergo structural and functional changes, known as cell adaptation. If the cells cannot adapt according to the changes in their microenvironment, they undergo cell injury. Cell injury could be reversible or irreversible. Virchow's cellular theory of disease, 1859: disease occurred due to abnormalities at the level of cells. The cellular response to stress may vary and depend upon the following: the type of cell and tissue involved, extent and type of cell injury. The causes of cell injury is divided into two: acquired and genetic. The first one is hypoxia. So what does hypoxia mean? Hypoxia mean reduce the amount of oxygen in the tissue, causing cell injury by reducing aerobic oxidative respiration. Second one is ischemia, means reduce blood flow. It can be arterial blood flow or it can be venous blood flow. Both can cause ischemia. Common poisons like arsenic, mercury, cyanide interfere with cellular metabolism. Injuries by microbes include infections caused by fungi, rickettsia, bacteria, parasites and viruses. Immunological cause. Always remember, immune system is very important for our body. It protects us from different harmful endogenous as well as exogenous agents, but the problem is sometimes the immune system find our own cells as enemies and they start to produce some immune response against our own cell, and that is the term known as autoimmune disease, and that can cause cell injury. Dietary insufficiency of proteins, vitamins and minerals can lead to injury at the cellular level due to interference in normal metabolic pathways. Physical agents, exposure of tissue to extreme heat or cold results in direct injury that is often irreversible, resulting in a pattern of coagulative necrosis. Genetic derangements result in a defect as severe as the congenital malformations associated with Down syndrome, caused by chromosomal abnormalities. Inborn error of metabolism arising from enzymatic abnormalities. Now we'll discuss the pathogenesis of cell injury, but before that, understand the role of ATP. Mitochondria is concerned with cell respiration and the production of ATP, which is responsible about important vital functions of the cell: cellular osmolarity, Na/K, membrane transport process, protein synthesis. Mitochondrial oxidative phosphorylation is disrupted first and leads to decreased ATP, and also cause the leakage of some pro-apoptotic molecules, some molecules that promote apoptosis, and that will result in cell injury. When the ATP is depleted or reduced, our body needs to generate ATP. So how will you generate ATP without oxygen? So by the process of anaerobic glycolysis. In this process, the amount of ATP produced will not be sufficient. However, there will be also some lactic acid produced in anaerobic glycolysis, and this will result in lower pH, and this will lead to clumping of nuclear chromatin. The next thing that will happen when we have reduced ATP is problem with the sodium pump. There will accumulate water as sodium, and water begins to accumulate inside the cell. The cells will swell up and result in cell swelling. What is reversible cell injury? It occurs when environment changes exceed the capacity of the cell to maintain normal homeostasis. But if the stress is removed or the cell withstands the assault, the injury is reversible. The biochemical and ultrastructural changes in the reversible cell injury are as follows, in which the first one is decreased generation of cellular ATP due to mitochondrial damage. Living cells require continuous supply of oxygen, further synthesis of ATP, and the ATP is derived from aerobic respiration or oxidative phosphorylation in the mitochondria, and to maintain the constant supply of ATP, cells may switch over anaerobic glycolysis oxidation, especially in the ischemic conditions as you can see in the flowchart. The second event is intracellular lactic acidosis. It mainly causes nuclear clumping. The third event is damage to plasma membrane pumps. So whenever there is damage to the plasma membrane pumps, there will be a hydrophobic swelling and other membrane change. If ischemia or hypoxia is for short period of time, the cell can be reverting back to its normal condition, which is known as RCI, reversible cell injury. There are 4 types of reversible cell injury: cellular swelling, fatty change, hyaline change, mucoid change. Cellular swelling is defined as accumulation as watery fluid in cells. Cells are incapable to maintain ionic and fluid homeostasis. Commonest and earliest form of cell injury. Common causes are bacterial toxins, chemicals, poisons, burns, high fever. The morphological changes during cellular swelling: cells are swollen, indistinct cell margins, and cytoplasm filled with eosinophilic granules, and cell borders might be grade-releasing granules. A severe form of cloudy swelling. It is the result of failure of energy-dependent ion pumps in the plasma membrane. Fatty changes is the accumulation of fat in non-fatty cells. It is also known as steatosis or fatty metamorphosis, is the accumulation of neutral fat within parenchymal cells. Here you can see that when the normal cell undergo with some fatty changes, manifested by appearance of lipid vacuoles in the cytoplasm, seen in kidney, heart and liver. Hyaline change, not a distinct chemical entity. Various histological or cytological alterations characterized by homogeneous, glass-like appearance in hematoxylin and eosin-stained sections. Bullet. It may be intracellular or extracellular. Intracellular hyaline colon, it's mainly seen in epithelial cells. Hyaline droplets in the proximal tubular epithelial cells. Excessive reabsorption of plasma proteins. Bullet. Hyaline degeneration of rectus abdominis muscle called Zenker's degeneration, occurring in typhoid fever. Zenker's degeneration, occurring in typhoid fever. Mallory's hyaline represents aggregates of intermediate filaments in the hepatocytes in alcoholic liver cell injury. Bullet. Nuclear or cytoplasmic hyaline inclusions seen in some viral infections. Bullet. Russell's bodies representing excessive immunoglobulins in the rough endoplasmic reticulum of the plasma cells. A few examples of extracellular hyaline change are as under. Hyaline degeneration in leiomyomas of the uterus. Hyalinized old scar of fibrocollagenous tissues. Hyalinized old scar of fibrocollagenous tissues. Hyaline arteriolosclerosis in renal vessels and hypertension and diabetes mellitus. Mucus containing mucin left parenthesis glycoprotein, secreted by mucus glands. In certain cellular injury, there is change characterized by accumulation of mucin in intracellular or extracellular loci, abnormal accumulation of excess amounts of various normal substances left parenthesis, water, lipid, proteins, pigments, abnormal substances left parenthesis, endogenous and exogenous. What is irreversible injury? Cell death is a state of irreversible injury. It may occur in the living body as a local or focal change. Autolysis, necrosis and apoptosis. The changes that follow it. Gangrene and pathological calcification. Damage to mitochondria is one of the hallmarks of irreversible cell injury and is usually a common feature of all injurious biochemical pathways that follow cell injury. Thanks for watching. Please like, share and subscribe my channel.